two reactions, first you need to know the criteria for distinguishing between these two. And these criteria you can use, for example, you can say one of the criteria, you may use a definition between light-dependent reaction and the light-independent reaction. But another criteria that you can use to differentiate between these two reactions, you can use also this location in the chloroplast. Therefore, location in the chloroplast also is another criteria. But again, you can use also the product. You remember the previous lesson, we were talking about light-dependent reaction. In light-dependent reaction, we saw some of the products which are formed there, which are very important for the next step of photosynthesis. You see, also the criteria that you may use is you may use the enzyme involved. We have light-dependent reaction and the light-independent reaction. All of these reactions are enzyme-controlled reactions. Therefore, in order to distinguish between these two, also you can use the enzyme involved. Also, another criteria that you may use the requirement for light reaction to occur, requirement for light-independent reaction to occur. Okay, let us start explaining this concept of light-independent reaction. Okay, light-independent reaction, actually it takes place in the stroma of the chloroplast. It takes place in the stroma of the chloroplast, but formerly this light-independent reaction was known as dark reaction. But nowadays, it is no longer known as a dark reaction because it is seen that there is some incorrect. Because when you say dark reaction, it means you mean that it occurs only in absence of light. But the recent research showed that this reaction can take place in presence of light and even in absence of light. Therefore, to be in correct ways, it's better you say light-independent reaction. Okay, this light-independent reaction, the overall purpose of this is to convert carbon dioxide from the atmosphere into carbohydrate. Therefore, how this process occur, how this conversion of carbon dioxide into carbohydrate occur, this is what we are going to explain here. Okay, light-independent reaction, this is enzymatic controlled reaction which was explained by a scientist known as Calvin Melvin. And that is why sometimes this light-independent reaction is also known as a C3 cycle or Calvin cycle. Why it is known as C3 cycle? Why it is known as Calvin cycle? C3 cycle is because of, according to Calvin Melvin, the first product which was isolated in this plant, it has a 3 carbon. Therefore, when you're talking about 3 cycle, it means the first stable product to be formed, it has a 3 carbon. And why we call it Calvin cycle? We call it Calvin cycle because of the scientist of this cycle. He was known as Calvin Melvin. Okay, this process of light-independent reaction, it involves 3 main phases. And these are, the first one, carbon dioxide fixation. A second one is reduction, or we call it reduction of phosphoglyceric acid. And the third one is regeneration Okay, these are three main phases of light independent reaction. We have carbon dioxide fixation, reduction of phosphoglyceric acid, and the last one is regeneration of carbon dioxide acceptor called the ribulose biphosphate. Ribulose biphosphate. Okay, let us go through these phases of light independent reaction. Let us see how carbon dioxide fixation occur, how reduction occur, and also how regeneration of this carbon dioxide acceptor occurs. Starting with first phase, carbon dioxide. Okay, carbon dioxide fixation. When you are talking about carbon dioxide fixation, as you have said, the main purpose of the light independent reaction is to convert carbon dioxide into a carbohydrate. Therefore, the first stage is carbon dioxide fixation. How carbon dioxide fixation occur? First of all, we are saying the carbon dioxide diffuses into the plant tissue through the stomata pores located in the leaves or stem of some plants such as cactus. Therefore, when this carbon dioxide diffuses into the plant tissue, it combines with one of the compound known as ribulose biphosphate, a carbon dioxide acceptor. Therefore, the carbon dioxide, when it diffuses into the plant tissue, it combines with a 5-carbon compound known as rubip. This is 5-carbon compound. The long form of rubip is known as ribulose biphosphate. Therefore, this is carbon dioxide acceptor. Again, after combining with carbon dioxide, it forms the product known as phosphoglyceric acid. Phosphoglyceric acid. But remember that this phosphoglyceric acid is stable product, the first stable product to be formed. But before the formation of this, there is when the one carbon dioxide is accepted by this carbon dioxide acceptor rubip, it forms first a unstable 6C compound. This 6C compound is unstable, therefore immediately break to form 2 molecules of phosphoglyceric acid. Therefore, it breaks to form 2 molecules of phosphoglyceric acid. But this reaction is catalyzed by the enzymes known as rubip carboxylase. Rubip carboxylase. Okay, therefore, this enzyme facilitates the formation of this product. This rubip carboxylase also it is known as rubisco. Rubisco. The long form of rubisco is ribulose biphosphate carboxylase OXJ. Okay, this is a long form of rubisco. Again, this is enzyme which help this first process of light independent reaction. We call it rubip carboxylase, or we call it rubisco. Why it is known as rubisco? This enzyme can fix both carbon dioxide and the oxygen. This enzyme can fix both carbon dioxide and oxygen. Therefore, depending on the concentration of this carbon dioxide and the oxygen, the rubip can act as a rubip carboxylase or rubip oxygenase. You see? Therefore, because here it fixes a carbon dioxide, therefore it is known as rubip carboxylase. Okay. Therefore, the phosphoglyceric acid, the short form of this, it is written as PGA, phosphoglyceric acid. Okay, the formation of phosphoglyceric acid marks the end of the first stage of light independent reaction. Therefore, after formation of phosphoglyceric acid, again, this phosphoglyceric acid is forwarded into a second stage of light independent reaction, and this is reduction. Reduction of phosphoglyceric acid, the second stage of light independent reaction. Okay, let us see how this reduction of phosphoglyceric acid proceeds. Okay, we are saying this process reduction, it requires energy, and this energy comes from the product of light reaction. Okay, before I'm explaining this, let me take you to one of the questions. Before explaining this concept, let us see some of the sample questions which we can use to explain this. Why it is important for light dependent reaction to proceed before light independent reaction? Why it is important for light dependent reaction to proceed before light independent reaction? Why is it important? During reduction phase, the reduction, it requires the product of light reaction. Therefore, products of light reaction such as ATP and the reduced NADP are the one which are used in this step. Therefore, it is important for light dependent reaction to proceed before light independent reaction, because without this light dependent reaction, the light independent reaction cannot proceed. Cannot proceed because of what? Because light independent reaction depend on the product of light dependent reaction. Okay, therefore here we have a phosphoglyceric acid, this PGA, as you say, this is stand for phosphoglyceric acid, is converted into another product, is converted into another compound known as phosphoglyceraldehyde, PGAL. Call it phosphoglycer. Okay, but this conversion, it requires energy. This conversion requires energy. Where does this energy come from? Where does this energy come from? As I have said, it is in this process where those products of light reaction are used, and those products are ATP and reduced NADP. Therefore, the main function of these two products, ATP, it supply energy, because for this process to occur, it requires energy, but also reduced NADP, it remove the oxygen from the phosphoglyceric acid so as to form phosphoglyceraldehyde. Principally, this PGA is carboxylic acid, and this PGAL is aldehyde. Okay, therefore this NADP, it reduces or it removes oxygen from PGA to form a PGAL. Okay, therefore in this question here, you can say we have ATP. When ATP is broken down, it releases energy. Therefore, it forms ADP plus inorganic phosphate. But also this reducing power. We have this NADPH2 to form oxidized NADP. P. You see, the long form of this NADP, this is nicotine amide, adenine dinucleotide. Phosphate. Okay, therefore now we have this phosphoglyceraldehyde. Again, as you have said, this is a 3-carbon compound. PGA, again, this is a 3-carbon compound. Again, this 3-carbon compound. Therefore, we have these 2 molecules again converted to form 2 molecules of phosphoglyceraldehyde. Okay, therefore the formation of phosphoglyceraldehyde marked the end of the secondary stage of light-independent reaction. After formation of this phosphoglyceraldehyde, again phosphoglyceraldehyde is sorted into the last stage of light-independent reaction. We call it regeneration of carbon dioxide acceptor, known as RUBIP. Okay, the third stage is a regeneration of carbon dioxide acceptor called the ribulose bisphosphate. As you said, this compound is 5-carbon compound. Short form of this is written as RUBIP, and this is 5-carbon compound. Okay, this 5-carbon compound, let us see how this 5-carbon compound is formed. How this 5-carbon compound is formed. The main event during regeneration of carbon dioxide acceptor actually is a rearrangement of carbon atoms. Rearrangement of carbon atoms. Therefore, PGAL of phosphoglyceraldehyde, again there is a rearrangement of carbon atom between sugar phosphate. Therefore, this PGAL, as you have said, this is a 3-carbon compound. 3-carbon compound. As you have said, in regeneration, what's happened here is rearrangement of carbon atom. Therefore, in the arrangement of carbon atom, phosphoglyceraldehyde is converted into a 5-carbon compound known as ribulose bisphosphate. Therefore, PGAL first it is converted into molecules known as RUP. This is 5-carbon compound is RUP, ribulose phosphate. Okay. Therefore, first the 3-carbon compound is converted into 5-carbon compound to form what we call a RUP. And again, this RUP is converted into ribulose biphosphate with the help of ATP from the light reaction. Therefore, ATP here is used, and the ADP okay. Therefore, this is 5-carbon compound with 2 phosphate. Therefore, ribulose phosphate, it means it have only 1 phosphate. After addition of phosphate from ATP, it gives the compound which has a 2 phosphate. Therefore, RUBIP, we call it RUBIP. This is carbon dioxide acceptor we are talking. Therefore, these 3 stages are the one which are involved in the whole process of light independent reaction. Let's take you back to some of the sample questions so that you can understand how this process can be explained. Let's see this question. Describe the process involved in conversion of carbon dioxide into sugar. Yes, describe the process involved in conversion of carbon dioxide into sugar. The key hint or the key point of this question, because we have said when you want to the main purpose of light independent reaction is to convert carbon dioxide into carbohydrate, sugar such as glucose. Therefore, this process, it means in this question, what you are supposed to describe here is the process of light independent reaction from the stage number 1 up to stage number 3. Carbon dioxide fixation, how does it occur? Reduction, how does it occur? And also regeneration, how does it occur? But also this question, you may be asked in other way, let's say with the help with the aid of diagram. Describe yes, same question. With the aid of diagram, describe Calvin cycle. With the aid of diagram, describe the Calvin cycle. Therefore, the answer will be based on the process of light independent reaction from the stage number 1 up to stage number 3. Okay, for this Calvin cycle to occur, the cycle must run 6 times. Because each turns, the cycle gives 1 carbon dioxide to form a phosphoglyceric acid. Therefore, for this cycle to occur, it must run 6 times. Okay, let's see diagrammatically how this Calvin cycle proceeds. Okay, as students, again the Calvin cycle can be explained diagrammatically as follows. Okay, but before I show you this diagrammatical explanation of Calvin cycle, you need to remember that for 1 molecule of glucose to be made, this cycle must run 6 times, because you remember the glucose, it has a 6 carbon. Therefore, for this cycle, or for 1 molecule of glucose to be made, the cycle must run 6 times. It means each times, or in each cycle, the cycle gives only carbon dioxide to phosphoglyceric acid so as to form a glucose. Let us see. As we have said, we have a carbon dioxide acceptor known as RUBIP. As we have said, this RUBIP is 5 carbon compound. When this 5 carbon compound accept carbon dioxide from the atmosphere, it forms unstable when 5 carbon compound accept this carbon dioxide, it forms a 6C compound. This 6C compound is unstable, therefore immediately break to form 2 molecules of phosphoglyceric acid. Okay, but as we have said, for 1 molecule of glucose to be made, this cycle must run 6 times. Therefore, let's assume that we have 6 molecules of RUBIP, 6 molecule of rubip. Therefore, when this 6 molecule of rubip accept a 6 molecules of carbon dioxide, it means it will form a 12 molecules of PGA times 6, or we can say 12 molecules of phosphoglyceric acid. So the enzymes, this reaction is catalyzed by the enzyme, and the enzyme which is responsible here, if you remember, we have said is a rubip carboxylase. Okay, after formation of this phosphoglyceric acid, mark the end of the first stage of Calvin cycle, which is, we call it carbon dioxide fixation, or sometimes we call it the reaction, we call it carboxylation. Therefore, this is first stage, call it carbon dioxide fixation, or we call it carboxylation. Carboxylation because the reaction involves addition of carbon dioxide into 5 carbon compound. Okay, as we have said, we have 3 main stages. After formation of this phosphoglyceric acid, another stage, or this phosphoglyceric acid, after formation of this phosphoglyceric acid is forwarded into the next stage of Calvin cycle, which is reduction. Therefore, this phosphoglyceric acid is reduced from phosphoglyceric acid. Again, if you remember, we have said this PGA is converted into PGAL. PGAL, phosphoglyceraldehyde. But we have 12 molecules of phosphoglyceric acid, and it is in this process where those main products of light reaction are used. You remember about those products of light reaction, which are ATP and the juicy NADP. Therefore, in this stage, we use these 2 products, ATP, so as to provide energy for conversion of this PGA into PGAL, but also, as we have said, this PGA is principally a carboxylic acid, but this PGAL is aldehyde. Therefore, what reducing power here it does is to remove the oxygen from phosphoglyceric acid to form the so-called phosphoglyceraldehyde. Okay, but we have 12 molecules of PGA. Again, in order to convert this PGA, again here we have 12 molecules of PGAL. Therefore, in order to convert this, again we need 12 molecules of ATP. Therefore, here 12 molecules of ATP. As we know, when this ATP is broken down, it releases energy. This energy is used for this reaction. 12 molecules, it forms a 12 ADP plus 12 inorganic phosphate. Again, because we have 12 molecules of PGA, we need 12 molecules of reducing power. Therefore, here we have 12 molecules of reducing power, which will produce 12 molecules of oxidizing power or oxidizing agent. See? Therefore, now the PGA has been converted into PGAL. And the formation of this PGAL, or we are saying this PGAL is the first carbohydrate to be formed during the process of photosynthesis. Yes. After formation of this PGAL, what next? This PGAL or phosphoglyceraldehyde, again, some of phosphoglyceraldehyde used to make a photosynthetic products such as glucose, amino acids, and other products. But remaining phosphoglyceraldehyde is used to regenerate the so-called carbon dioxide acceptor known as ribulose biphosphate. Yes, therefore, after formation of this, some used to make photosynthetic product, the remaining PGAL are used to regenerate a carbon dioxide acceptor. But keep in mind that in these 12 molecules of PGAL, only 2 molecules are used to make a photosynthetic product. The remaining 10 molecules, they are used to regenerate the so-called ribulose biphosphate. Therefore, from here we have this PGAL are 2 molecules separated from the cycle. You see, we have these 2 molecules. PGAL is used to make sugar, for example, glucose. And this is because we have said this PGAL is 3-carbon compound. Again, this is 3-carbon compound. Therefore, we know that glucose has a 6 carbon. Therefore, this PGAL, 3 carbon, another PGAL, 3 carbon, therefore total number of carbon 6. Therefore, it will be used to make the so-called glucose. The remaining 10 molecules, you see, the remaining 10 molecules, they are used to regenerate rubip. How this regeneration occur? It is there, we have said. Majority of the PGAL is forwarded into the last stage of Calvin cycle, which is regeneration of carbon dioxide acceptor. Yes, this PGAL first, during the main event, during regeneration, you need to remember, the main event during regeneration is we are talking about the rearrangement of carbon atom between a sugar phosphate. Therefore, what happened is only rearrangement of carbon atom between a sugar phosphate to convert 3 carbon compounds to form 5 carbon compound. As you can see here, this PGAL is 3 carbon compound, but we need to synthesize 5 carbon compound. How this proceeds? We are saying these 10 molecules first is converted into another compound known as RUP. And this is if you remember, we have said this RUBIP stands for ribulose biphosphate. It means this 5 carbon compound has 2 phosphate. But this RUP stands for ribulose phosphate. it means this compound has only one phosphate. Again, this rubip carboxylase is a ribulose bisphosphate carboxylases. Okay, the group has been formed. You see, this is 5-carbon compound. As we have said, 3-carbon compound to form a 5-carbon compound. Therefore, here we need to convert 10 molecules of 3-carbon compound to form a 60 molecules of 5-carbon compound. As you can see here, we have 10 molecules, but in each molecule it has a 3 carbon. Therefore, it has, the total number of carbon, when you take 10 times 3, we have 30 carbon. Again here, we have 60 molecules, but each molecule has a 5 carbon. Therefore, 60 molecules with 5 carbon, again, the total number of carbon is 30. After formation of this group, this group is converted into rubip. Rubip, this is again a 5-carbon compound, but the difference between this and this, rup, it has only one phosphate, rubip, it has a 2 phosphate. Therefore, to convert rup to rubip, we need to have phosphate. Where does this phosphate come from? From one of the product of light reaction, which is ATP. This ATP, it is broken down. You see? We need ATP. When this ATP is broken down, again, it provides a phosphate. Therefore, it forms the so-called ADP. The phosphate already added into this compound so as to form bisphosphate, 2-phosphate. But again, because we have 60 rup, we need to have a 6 ATP because each molecules need phosphate. Therefore, we need to have 6 ATP, and this 6 ATP again will produce a 6 ADP. Okay, therefore, this is how Calvin cycle is explained diagrammatically. It's called Calvin cycle. As you can see, this is the first stage, carboxylation. This is a second stage, reduction phase. And this is third stage, regeneration. Okay, up to now, I think you are able to explain the Calvin cycle diagrammatically with the help. For example, if the question is, with the help of diagrams, describe Calvin cycle. You should be able to explain each step from carbon dioxide fixation to the regeneration of carbon dioxide acceptor. So, my students, let me conclude our session of today. We were talking about a light independent reaction, but in light independent reaction, we need to know the main event during light independent reaction. As we have seen, the Calvin cycle is a main event during light independent reaction. Therefore, the things to remember, those 3 stages, but not only to remember those 3 stages, how these stages are explained. How these stages are explained. Therefore, you should be able now to explain carboxylation, reduction, and the last one, which is regeneration. I think up to now, from my explanation, you should be able now to answer many questions from different books. And the good reference, you can use a biological science. It has many questions about this part, but also understanding biology, again, it has many questions about this part. Even the book of Thai, you see, there are so many questions from this part. Thank you so much for listening, but I'm inviting you to join me to the next session, which we shall talk about the issue of C3 plant and C4 plant. As you have seen here, there are some of the plants during photosynthesis, the first product to be formed has a 3 carbon, but there are some of the plants in which the first product to be formed has a 4 carbon. Therefore, from this, we can be able now to explain about the issue of C3 plant and C4 plant. Thank you so much. Bye-bye.
Kipindi cha Darasa Online kimeletwa kwenu na Chuo Kikuu cha Sauti Mbeya.